Autophagy's role in cancer is often viewed through the lens of individual tumor cells, either as a mechanism that suppresses early tumor development or as one that aids survival in advanced disease. But this focus overlooks a crucial dimension: the tumor microenvironment, a complex ecosystem of non-cancerous cells, extracellular matrix, and signaling molecules that collectively influence tumor behavior. Within this environment, autophagy doesn't operate in isolation. In fact, autophagy in stromal cells such as fibroblasts, immune cells, and endothelial cells can profoundly affect tumor progression, sometimes in unexpected and paradoxical ways. One of the more striking findings in recent cancer research is that autophagy in the surrounding non-malignant cells can actually fuel tumor growth by providing metabolic support. In nutrient-deprived tumors, especially those with poor vascularization, cancer cells often rely on the breakdown products of neighboring stromal cells to survive. Stromal autophagy contributes to this by degrading intracellular components and releasing amino acids, fatty acids, and other nutrients into the microenvironment. These nutrients are then taken up by cancer cells to sustain proliferation, resist apoptosis, and even evade certain therapies. In this way, autophagy in ostensibly normal cells becomes a metabolic engine for malignancy, a process sometimes described as autophagic feeding. Cancer-associated fibroblasts, CAFs, in particular, demonstrate this effect. When activated in the tumor microenvironment, these fibroblasts often show high levels of autophagy and secrete nutrient-rich vesicles that support tumor metabolism. Similarly, autophagic activity in immune cells or endothelial cells can modulate inflammation, angiogenesis, and immune surveillance, sometimes dampening the immune response and allowing tumors to progress unchecked. This reprogramming of stromal cells turns what might otherwise be a hostile environment into a supportive niche for cancer growth. Yet autophagy in the tumor microenvironment is not exclusively tumor-promoting. In certain contexts, it helps restrain cancer by supporting immune cell function, clearing damaged matrix components, and limiting inflammation. For example, autophagy in antigen-presenting cells enhances tumor antigen presentation and improves T cell activation. In these scenarios, autophagy contributes to a more immunologically active and anti-tumor microenvironment. Whether autophagy is ultimately protective or permissive depends on the balance of cell types, the stage of the tumor, and the surrounding metabolic conditions. This duality complicates efforts to target autophagy therapeutically. Inhibiting autophagy in cancer cells might slow growth or sensitize tumors to treatment, but systemic inhibition could impair immune function or disrupt supportive autophagy in healthy tissues. Likewise, attempting to boost autophagy to enhance immune responses or prevent early tumor formation could inadvertently provide more metabolic fuel to cancer cells through stromal pathways. The challenge is to develop therapies that are spatially and temporally precise, capable of distinguishing between autophagy that sustains normal tissue function and autophagy that inadvertently supports malignancy. Emerging research into cell-specific autophagy modulators and tumor microenvironment profiling may eventually make this level of precision possible. Techniques such as single-cell RNA sequencing and spatial transcriptomics are beginning to map out the heterogeneity of autophagy-related gene expression in tumors, offering clues to when and where interventions might be most effective. Combination therapies that target both cancer cells and their stromal support systems are also being explored, particularly in cancers known to depend heavily on metabolic cooperation. Ultimately, autophagy in the tumor microenvironment exemplifies the complexity of cancer as a systemic disease. It's not just about rogue cells multiplying unchecked, it's about the intricate and often cooperative interactions between those cells and their neighbors. Autophagy, with its capacity to either restore balance or shift the equilibrium toward growth and survival, acts as a key modulator of this ecosystem. To understand and manipulate cancer effectively, we must consider autophagy not as a singular process, but as a contextual force, capable of reshaping the environment in which tumors live, thrive, or die.